for the parasitoid Leptomastic dactylopy. The first parameter is the direct exposure and effect of residuals on adults. To perform that, female and male of the parasitoid will be isolated with treated plants with our products. And we will check the survival and dead parasitoids for 72 hours. The second parameter is the parasitization rate of females exposed to the residuals, and this will be done with the female survivals from the previous experiment. So the female survivals will be isolated with infested plants with Planococcus citrinas. After a couple of days, the mummies formed will be isolated, and we will check for the parasitoid emergence and sex ratio. The third parameter is direct effect on cocooned pupa. To perform that, we need infested plants with Planococcus citri, and we will isolate with this plant female and male of parasitoid. And after the formation of the mummies, we will treat our plants, and we will isolate the mummies in glass vials, and we will check the adult emergence rate, time required for emergence, and sex ratio. The fourth parameter, which is for us the most important, is the effect on the host searching of the behavior. To do that, so mated and unmated females will be put in a Y olfactometer, and we will allow each individual to choose between these combinations. So we have infested versus non-infested, treated versus non-treated, and treated versus non-treated. And the choice made, time spent in each arm for each individual will be recorded. The secondary experiments will be performed on the lady beetle, Cryptolemus montrosieri. So here we will perform the experiment on larvae and on adults. So the effect of products and their screening on larvae and adults, as I mentioned. So we will take the larvae or the adult. We will isolate this with treated plants with our products, infested with the third instar NAS for 24 hours. After 24 hours, we will transfer the adult or the larvae on infested plants, but not treated. And we will evaluate the daily consumption rate and the survival and check fertility for females, and this for 72 hours. The second parameter is the effect of direct treatment on pupa. And here also, we will isolate a couple of the lady beetle, male and females, with infested plants with Planococcus citri. After a couple of days, we will treat the plants presenting the pupa, and we will isolate the pupa inside glass vials, and we will check the adult emergence. So the expected results are the efficacy of essential orange oil and Clitoria ternatia extract in controlling Planococcus with all its stages, crawlers, nymphs, adults, and on Aunediella auranti. And the assessment of the acute toxicity and sublethal effect of this product on Citrus mealybug, natural enemies, so Leptomastic dactylopy and Cryptolemus montrosieri. So this is the timeline. So we are here, and coming soon the lab experiments and field trials. These are the references cited in the presentation, and thank you for your attention.
Thank you very much, Sonia. Monsieur Hippolyte, you want to start?
Yeah. Be quiet, no problem. I will not ask something.
Don't be nervous.
Don't be nervous. A good presentation, I appreciate it. Only two objectives that looks feasible. Yeah, I lose myself when you talk about the different treatment you have to do, but I think it's fine. The only thing that I suggest, but maybe, is for this preliminary presentation, how many plants you will use, how many treatments, how many repetitions. This will be fine to present maybe in next seminar.
If you want, I can give an answer because I didn't put it on the presentation. Yeah. For example, it depends on the parameter, but we are 35 plants by experiment, and in some cases more than 35. It depends on the parameter.
Yeah, because for statistical significance.
Yes. And sometimes we are working on the pupa, so we will take like 30 pupa. So it depends on the experiment. And the variety that we will use is Valencianate for those models.
Valencia.
So you already have this methodological
Yes.
information.
Good. Okay, thank you.
You're welcome.
Me, it's very clear, so congratulations. No technical questions.
Okay, Eva?
Also for me, congratulations. Very important, interesting work. I hope that you will not overload yourself with so many experiments, because it's just to remind that it's a master thesis work in a short time, not a PhD.
Yeah, very clear, very nice presentation, lots of experiments. Also, I share with the Professor